because of the mechanisms of the defense of intrinsic to the cells, the animals recover because this is like, in fact, we.
Sorry.
And also because we performed only 3 injections. I think if we performed more injections maybe the animal not recover. I don't know. We did not measure that, but it's possible.
Okay. Do you think that this mitochondrial damage, that obviously you have a release of sting, but you have a great release of oxygen reactive radicals, and do you think that this mitochondrial damage probably surpass the inhibition of sting, and other mechanisms can is working in it? Probably not, because you have a great release of ROS in this, and if you use an inhibitor of sting or gas, and for example, antioxidants, probably you can surpass this and potentiate the effect. Do you think that's possible?
Yeah, I think that it's ROS. It's evident that ROS it's produced in response to cystatin treatment, and I think yes, it can potentiate the effect. Even though the cell have antioxidant mechanism to avoid this increased production of ROS, I think that it can potentiate the mitochondrial damage, leading to more mitochondrial damage, leading to more extravasation of mitochondrial DNA to the cytoplasm, and then activating sting. That is a possibility.
Yeah. My last question is, it's a curiosity. This damage to mitochondria in a sting, this damage to mitochondria caused by cystatin occurs in any cells or more in neurons or in ganglions?
Not only in the cells of the dorsal root ganglia. I believe that cystatin can cause mitochondrial damage in many cells. For example, there was a study on the literature that demonstrates similar effects on the cells in the context of the acute kidney injury. So, for example, cells of the kidney also have mitochondrial damage. So I think this is not restricted to the dorsal root ganglia, even though the dorsal root ganglia is not protected by the brain-blood barrier. Other cells would also have mitochondrial damage.
So to use the sting inhibition, maybe it's great not for leukopen, but for other damages. Yeah. Thank you, Fabio. Do you have any question, Calixto? Você está mudo. You are mute.
I have just one comment and concern. I noticed that very nice talk, this one. Clear, good result. But use group of 3, 4, 5, 6, 7, 10 animal per group or experiment. I am concerned about that because the crisis of reproducibility of data today showed that very important statistical analysis you find in 3 or 4 animals not justify the power because the number is so small. What do you think about that?
Oh, can you repeat more slowly?
No, because you use different animal, the sample you use. There is no how you calculate the size of the sample. But you have different size of the sample for statistical analysis. So my question is, The result is very good, very strong, but I think that the number of experiments should be improved in some cases, not 3, not 4, not 5, but more. It's possible the same. I know that the problem is knockout animal. I know that, but for heiferi, it is not justified because heiferi is heiferi. What do you think about the 5 animals to make statistical analysis?
Yeah, you are talking about the behavior test.
More behavior, but also in molecular biology as well.
Yeah, I think that we have a facility here in Ribeirão Preto Medical School that provides us the knockout animals. So we use this quantity of animals because to provide a strong result, to provide a strong phenotype that it's very clearly so. As clearly we have the results, I think it's important because we don't have to repeat more and more to observe the same phenotype. So I think that's it.
The question is not the reproduce, the question is statistical power. If you don't have statistical power, there is no way to justify it.
Okay, I don't know.
I'm fine. Okay, but it's just a concern.
Okay, I think it's very valuable this consideration, and we're going to took that on mind in our next evaluations and meetings. Thank you.
I don't know if Juan Saavedra, Dr. Juan Saavedra is online. Gabriela, do you know if Dr. Saavedra is online?
No, he's not online right now. I'm sorry.
Okay. Fábio, I should still call you Thiago. And thank you very much. It's a nice presentation. And let's go to call our last candidate is Viviano. Viviano is here. Viviano Neves, and his work is entitled Matrix Metal Proteinase and NP2 Proteolysis, Type 1 Collagen, Collagen 1, and contributes to focal adhesion kinase FAC activation and vascularized muscle cells proliferation in aorta acute hypertensive wrecks. His co-authors is Blanca de Mello, Rodrigues, Rocha, Parente, Dr. Rita Tostes, and Castro, and Michele Mazon de Castro. Thank you. From the Department of Pharmacology, Ribeirão Preto Medical Faculty, Universidade de São Paulo, USP. Please, Viviano, the microphone is yours.
Hello, everyone. My name is Viviano. I am PhD student at Ribeirão Preto Medical School in the Department of the Pharmacology. Well, my work in trial. Matrix metalloproteinase type 2 proteolyze type 1 collagen and contribute to focal adhesion kinase activation and vascularized muscle cells proliferation in aorta of acute hypertensive wrecks. Well, first, let me talk about hypertrophic remodeling. In this picture, you can see aortic vessel in two conditions, in normotensive condition, in hypertensive condition. In hypertensive condition, the cell starts to express more matrix metalloproteinase type 2. To simplify it, I call MMP2. Different stimuli for mechanical stimulus or ROS or angiotensin type 2 tend to induce the small muscle cell to express MMP2. The MMP2, once it's created out the cells, can degrade the structure of extracellular matrix such as collagen, elastin, and thereby favoring the cell phenotype change and induce the cell migration and proliferation. As a final result, there is an increase the thickness of the medial layers of the vessel characterized by hypertrophical remodeling. Consequently, hypertrophic remodeling increases the risk of development of the cardiac disease. Well, the collagen present in the matrix extracellular participates the vessel structure as well as stiffness. Collagen can also participate the silent vapor as shown on the picture review. For example, type 4 collagen, type 1 collagen, but I will give you the POX and the type 1 collagen because it's the most abundant in the extracellular matrix. The type 1 collagen can activate the intervening receptor by a specific site of the amino acid called IgG. IgG is arginine, lysine, and asparagine. The collagen degradation by MMPs, the product of the degradation can bind to intervening receptor and activate the FAC by phosphorylation, thus induce the produce a cyclic cascade that will result in a cell migration and proliferation. Well, my hypothesis, my work is in acute hypertension, the vessel express more activation of MMP2. The MMP2 can lead to degradation of type 1 collagen. It is which increase the interaction with intervening receptor that will lead to FAC activation. And that will induce the cell proliferation, that the final result they produce the hypertrophic arterial remodeling. Well, for methodology, I work with hypertense model animal by 2 kidneys, 1 kidney model. This protocol was performed in 7 days. Sorry about this running. At the 30 day, from the 30 day onto the 7 day, the animals was created by doxycycline. Doxycycline is a MMP2 inhibitor, and then MMP2 inhibitor is the animals were treated with toxin by oral gavage. After the 7 days, the aorta was collected. After the 7 days, the animals were euthanized and the aorta were collected, and fully protocol was performed. Western blot for FAC, integrin receptors, and MMP2. Hydroxyproline for collagen assay degradation, immunofluorescence Ki-67, morphology, zymography, and 2A ANOVA for statistic test was performed. Well, The plethysmography data show an increase in the blood pressure in hypertensive group compared to the control the sham group. And the treatment with doxycycline did not reduce the level, did not reduce the blood pressure, systolic blood pressure. However, we evaluate the levels MMP2, increasing in hypertensive group, and increase the activity and hypertensive group, and in the zymographene gel, increase the active MP2 72 kilodalton and 64 kilodalton. 64 kilodalton is a MP2 is a form without a prepeptide. The treatment with doxycycline reduce the MMP2 levels and reduce the activity. Well, when we evaluate the type 1 collagen, once MMP2 was increased, type 1 collagen, you can see type 1 collagen degradation in hypertensive group, and you can see 18 kilodalton possible collagen products, and you can see high level hydroxyproline in hypertensive group animals. And the treatment with doxycycline maintain the levels of the type 1 collagen. You reduce the degradation of the collagen. We did not detect any difference in the quantification of the integrin receptor, except for beta-3 integrin decrease in the hypertensive group. And however, integrin in hypertensive animal is maybe overactivated because the fat phosphorylate is increased in the hypertensive group, and the treatment with the doxycycline reduce the fat activation or fat phosphorylation. Because of the once the fat was activated, we observed the cell proliferation in hypertensive group, and the treatment with doxy reduced the cell proliferation. As a final result, this activation, the fat pathway, induces hypertrophic arterial remodeling in hypertensive group. When evaluate the measure for the rate measure volume and the cross-section area. To also evaluate the cell migration and the fat activation, we work a 7R5 cell, a 7R5 cells. It is immortal cell line from the rats in the main medium. We work with a control fetal bovine sorrow, 2% fetal bovine sorrow is rich in MMP2. We stimulate the cells with the type 1 collagen, and ARP and DOX. DOX is a MMP2 inhibitor. PNG is a fat phosphorylation inhibitor. I work with two, ARP inhibitor and stimulation two with collagen. And the same with PNG, fat phosphorylation inhibitor, more stimulant of type 1 collagen. This protocol was performed in 24 hours, and I repeat the same assay, but for evaluate the cell migration. And in one assay, I did a risk in the middle of the plate, the well from the plate, for evaluate the migration. Finally, I take a picture in 0 hours, 6 hours, and 24 hours. Art is NMP2 inhibitor. When I collect the supernatant, we observe decreased NMP2 activity in cells treated with the ART. When collect the supernatants and precipitate the type 1 collagen stimulus, we can see the ART and DOX preserve the bands of the pro-collagen and type 1 collagen. Different from the control, has a lot of MMP2 activity, it does reduce the collagen levels. Well, when you see the type 1 collagen stimulate the FAC phosphorylation, and MMP2 inhibitor ART and DOX with collagen stimulus decrease the FAC phosphorylation. These results show that the importance of the degradation product from collagen, type 1 collagen, induce the activation of the FAC mechanism. Well, when I see the cell migration, you can see and the delta time, 6 hours, 18 hours, and 24 hours, you can see the collagen stimulate the cell migration in delta time 8 hours and 24 hours. In the treatment, if NMP2 inhibitor DOX and ART in 18 DOX ART and collagen DOX, you can reduce the cell migration. You can see in 24 hours for call on more doxycycline too. And when I use the FAC inhibitor, we can see reduce the cell migration in delta time in 18 hours and 24 hours. In conclusion, increase NMP2 expression and activity may be associated with COL1 degradation, which activates FAC and induces vessel smooth muscle cell proliferation and hypertrophic remodeling in early stage of hypertension.
Thank you, Viviane. I don't know if Dr. Juan is online. Gabriela?
Yes, he is.
Yeah. Please, Juan, if you want to begin the discussion. Juan, if you begin, we can begin. Yeah? Unmute, please.
I think he's trying, just the internet connection is not so good right there.
Yeah. Meanwhile, Kalish, do you want to ask anything?
No, I am fine.
Fine, okay. While Dr. Huang trying to do, thank you for to connect again. Thank you for your presentation. It's a very interesting work. And I have so your conclusion is that once MMN2 is activated, it cleaves collagen, and you have peptides produced that will activate alpha-1, alpha-2, beta-1 integrins, and then you have the activation of FAC and all the signaling throughout. Do you have, I don't know if I missed, but Did you use any alpha 1, alpha 2 integrin blocker?
Yeah, I didn't use, but
sorry, but your sound, it's very unclear.
Well,
so
you can hear me?
Yeah.
Well, I didn't use any integrin blocker, but in the I think in the next experiment I used the integrin block receptor for induce more be more representative for explaining that the intervene is activated by the degradation, the cleavation type 1 quality.
Okay. Juan, are you able to connect? Juan? And did you try to use any kind of MEMP2 inhibitor to even in vitro to inhibit?
Yes, I use MEMP2 in vitro. Is ARP. ARP is a specific inhibitor for MEMP2.
It's not so clear for me.
Sorry, I use the ARP. ARP100 is a specific inhibitor, and I see the same response in vitro compared to doxycycline.
Oh, okay. I did, yes, use ARP. Okay. I don't know if Juan.
I'm sorry about that because it was raining here. It's making noise. Sorry.
Yeah. And I think that you speak very, very close to the microphone. Juan, are you able to connect? Gabriel?
I think it's his connection. I mean, he is here. I'm sure he's trying to open his microphone, but for some reason he's not able to. But he's hearing us all the time. So
okay. Juan, if you want to type any question to Viviane in the chat. I don't know if I have time.
There is no more online.
Huh?
There is no more online.
It's frozen.
Yeah.
Okay.
Oh,
sorry about that. So if you have to compare with inhuman hypertension, what do you call acute hypertension prices in human compared to your model in rats?
Well, I think so. It's very important to know that. Are you hear me?
Yeah.
Okay. It's very important to know that in hypertension treatment, sometimes some treatment did not reduce MEMP2 levels. Well, when I use the MEMP2 inhibitor, it's very good when associated with hypertensive treatment. Well, and in my research, it's very important in acute hypertension because because when occur when in the later or chronic hypertension, the cell the vessel the vessel does produce the type the turnover from collagen. If this turnover change the characteristic of the collagen, and the MMP2 sometimes cannot break more the collagen because the turnover. And in my experiment, it's very important in acute hypertension.
Okay. Thank you, Viviane. I'm sorry that Dr. Juan couldn't ask you any question. So I thank you for your presentation. And Juan, you want to ask any question?
Regarding the last presentation.
Yeah, please.
Yes. Yeah, I wonder if there's any relationship to your findings with angiotensin II, which is a very important factor for remodeling, and whether this model of acute hypertension with chronic hypertension, for example.
Yes. In my experiment, it's important in acute
hypertensive patient.
Yes. In my experiment, it's very important in acute hypertension because increase the level of NMP2 levels, and this NMP2 activity can induce the proteolysis of the type 1 collagen. When occur a chronic hypertension, in our chronic hypertension occur collagen turnover. In this turnover, change the characteristic of the characteristic collagen characteristic, and NMP2 cannot more affected in this process.
Are you satisfied, Juan? Well, Marco Aurelio, I think that's...
Yes. Thank you all. The José Ribeiro do Vale Award is an annual SBF event sponsored by Biolab Pharmaceutical, aiming to stimulate our trainees, master's degree, PhD, from Brazil and Latin America, interested in developing the skills necessary for a fruitful scientific career. The award evaluating committee will now rank the candidates, and I will announce the winners during the closing session of our annual Congress tomorrow afternoon. Dear Professors Fidalgo, Saavedra, Calixto, dear candidates Tiago, Julie, Aurilene, Fabio, and Viviano, thank you all for this very interesting symposium. We are also, of course, grateful to our partner Biolab, and looking forward to keeping our partnership next year. Thank you for attending this session. We have an e-poster session ahead of us immediately after, so see you then. Bye-bye. Thank you all.
Thank you for all. Congrats for all. Marco, nós vamos nos reunir agora ou mais tarde?
Licença, a gente tem que finalizar esse link aqui por conta do e-poster, tá? Então, vou pedir licença aqui e vou autorizar a Softalisa a fechar o link.
Tá.
Ok.
Tá, então eu combino aqui com... I arranged a meeting online with you both, Calixto and Juan. Marco Aurelio wants to participate too?
No, I think you can sort it out. All right?
Okay.
And
okay.
let me know the result. Okay.
I send you Calixto and Saavedra